Hello, good morning friends. Welcome to our channel LabTech World. Myself Kishore Waghani, and today we are going to discuss 7th part of HPSSC, that is Himachal Pradesh Staff Selection Commission Lab Technician Exam question paper 2022. Before starting today's MCQ, I would like to request, if you are visiting our channel for first time, then please subscribe our channel LabTech World and press the bell icon to enable all notifications for our next upcoming videos. Let's start today's MCQ. Question number 114. Normal values of plasma total protein varies between option A 3 to 4 grams per deciliter, B 6 to 8 grams per deciliter, C 10 to 12 grams per deciliter, D 14 to 16 grams per deciliter. Fact available for this question, it is the normal range of total protein levels in the blood serum is 6 to 8 grams per deciliter. Out of these 6 to 8 grams, albumin makes up 3.5 to 5 grams per deciliter, and rest of the total protein is total globulin. So we can say from the given information, it is 6 to 8 grams per deciliter. Option B will be the correct answer for this question. Move to the next question number 122. Ketosis is associated with which disease? Option A, nephritis, B, diabetes mellitus, C, edema, D, coronary artery disease. Fact available for this question, it is normally insulin helps sugar to enter in your cells. Without enough insulin, your body cannot use sugar properly for the energy purpose. This prompts the release of hormones that break down the fat as a fuel, which produces the acid known as the ketones, very well known acetone, acetoacetate, and beta hydroxybutyric acid. Excess ketones build up in your blood and eventually spill over into the urine. This condition is associated with the 3 to 4 days of fasting or type 2 diabetes mellitus. So sometimes it is also known as the ketosis or diabetic ketoacidosis. So we can say it is diabetes mellitus that is associated with the ketosis. So option B will be the correct answer for this question. Move to the next question number 123. Coenzymes combines with option A, proenzyme, B, apoenzyme, C, holoenzyme, D, antienzyme. One of the repeatedly asked question. Fact available for this question, it is coenzymes participate in the catalysis when they binds to the active site of the enzyme called as the apoenzyme and subsequently form the active form of the enzyme that is known as the holoenzyme. So we can say from the given information, it is coenzyme combines with the apoenzyme to form a holoenzyme, that is the active form of the enzyme. So option B, apoenzyme, will be the correct answer for this question, to which coenzyme bind to form a holoenzyme. Move to the next question number 124. Which of the following serum enzyme rises or elevated in the myocardial infarction? Option A, creatine kinase, B, SGOT, C, LDH, D, all of the above. It is also one of the repeatedly asked question. Fact available for this question, it is cardiac enzymes, also known as the cardiac biomarkers, includes myoglobin, troponin, and creatine kinase. Historically, lactate dehydrogenase, that is LDH, and aspartate aminotransferase, that is AST, was also used as a cardiac biomarker, but it is non-specific one. So we can say it is all of the above. That means creatine kinase, SGOT, as well as LDH, all are the used as a enzyme that are raised in the myocardial infarction. So option D, all of the above, will be the correct answer for this question. Move to the next question number 125. The normal serum level of thyroxine or T4 is option A, 2 to 4 microgram per deciliter, B, 5.5 to 13.5 microgram per deciliter, C, 14 to 20.3 microgram per deciliter, D, 20 to 25 microgram per deciliter. Fact available for this question, it is thyroid hormones are the two hormones produced and released by the thyroid gland, namely triiodothyronine and thyroxine. They are tyrosine based hormones that are primarily responsible for the regulation of metabolism. T3 and T4 are partially composed of or combined with the iodine. A deficiency of iodine leads to decreased production of T3 and T4, enlarges the thyroid tissues, and will causes the disease known as the goiter. Talking about the normal values of thyroid hormones, that is T3 and T4, a normal T3 level might be somewhere between 100 to 200 nanograms per deciliter, while a normal T4 level falls between 5 to 12 microgram per deciliter. So we can say from the given information, for T4 it is 5 to 12 microgram per deciliter. So option B, 5.5 to 13.5 microgram per deciliter will be the most appropriate answer for this question. Move to the next question, number 126. Protein bound iodine is blank bound to protein. Options are A, iodine, B, thyroid hormone, C, thyroxine, D, triiodothyronine. Considering this question as a grammatical question, option A, iodine, will be the correct answer for this question, as protein bound iodine is iodine bound to protein. But as per the official answer key, they have given option B, thyroid hormone, as the correct answer for this question. Thyroid hormone includes thyroxine, that is T4, and triiodothyronine, that is T3. So it is difficult for me to explain the question as well as the correct answer for this question. So we have modified this question as iodine bound protein is blank. Options are A, thyroglobulin, B, TSH, C, TRH, D, thyroid hormone. Fact available for this question in form of figure, where you can see the iodine molecules when enter into the cell membrane. After activation, they will combine with the tyrosine produced from the Golgi apparatus to form a diiodotyrosine and monoiodotyrosine, which is further linked together to form a triiodothyronine and tetraiodothyronine or thyroxine, T3 and T4, which is further released into the bloodstream to reaches the peripheral tissues. So we can say from the given information, it is thyroid hormone or T3 or T4 that combines with the active form of the iodine. So we can say from the given information, it is thyroid hormone that is the iodine bound protein. So option D, thyroid hormone, will be the correct answer for this question. Move to the next question, number 127. The predominant cation of the plasma is option A, sodium, B, potassium, C, calcium, D, magnesium. Fact available for this question, it is the predominant cation or positively charged ion of the plasma is sodium, an ion that occurs within the cell at much lower concentration. Because of the effect of sodium on osmotic pressure and fluid movement, the amount of sodium in the body is an influential determinant of the total volume of the extracellular fluid. So we can say from the given information, it is sodium that is the predominant cation or positive charged ion of the plasma. Option A, sodium, will be the correct answer for this question. Move to the next question, number 128. Microscope was invented by option A, Ronald Rose, B, Robert Koch, C, Antony van Leeuwenhoek, D, Louis Pasteur. One of the easiest questions we have already covered in our MCQ series. Option C, Antony van Leeuwenhoek, will be the correct answer for this question. Move to the next question, number 129. Mycobacterium tuberculosis was discovered by option A, Louis Pasteur, B, Robert Koch, C, Lord Lister, D, Edward Jenner. Fact available for this question, it is also one of the repeatedly asked and the easiest question. Dr. Robert Koch was pivot figure in the golden age of microbiology. It was the German bacteriologist who discovered the bacteria that causes the anthrax, septicemia, tuberculosis and cholera, and his method enables others to identify many more important pathogens. So we can say from the given information, it is Robert Koch discovered the Mycobacterium tuberculosis, the causative agent of the tuberculosis. Option B will be the correct answer for this question statements. Move to the next question, number 130. All of the following pathogenic bacteria fulfills Koch's postulates except option A, Treponema pallidum, B, Yersinia pestis, C, Bacillus anthracis, D, Helicobacter pylori. One of the most important question, and the fact available for this question, it is Koch's postulates are as following. There are the 4 statements given by the Robert Koch that every pathogenic bacteria or organism should fulfill, and these 4 statements are as follow. Number 1, the microorganism must be found in abundance in all organisms suffering from the disease but should not be found in healthy organism. Second, the microorganism must be isolated from a diseased organism and grown in pure culture. And third one is the cultured microorganism should cause disease when introduced into a healthy organism. And last, number 4, the microorganism must be re-isolated from the inoculated or diseased experimental host and identified as being identical to the original specific causative organism or agent. So these all are the 4 statements known as the Koch's postulate. Talking about the exceptions, it is already widely accepted that some species of bacteria that causes disease, despite the fact that they do not fulfill the Koch's postulate, since Mycobacterium leprae, causative agent of the leprosy, and Treponema pallidum, causative agent of the syphilis, cannot be grown into the pure culture medium. That is the number 2 Koch's postulate. So it is from the given information we can say it is Treponema pallidum that does not fulfill the Koch's postulate. So option A, Treponema pallidum, will be the correct answer for this question. Move to the next question, number 131. Correct order of gram staining is option A, gentian violet, iodine, carbolfuchsin, B, iodine, iodine, gentian violet, carbolfuchsin, C, carbolfuchsin, iodine, gentian violet, D, carbolfuchsin, gentian violet, iodine. One of the easiest question. Fact available for this question, it is bacteria that retains the initial crystal violet stain, that is the primary stain, are said to be gram positive, where those that decolorize with the methanol or decolorizer and stains with the carbolfuchsin or safranin as a counter stain are said to be gram negative. So we can say it is carbolfuchsin can be used as a counter stain. So correct order of the gram staining should be gentian violet or crystal violet, then iodine, and lastly it should be safranin or carbolfuchsin. So option A will be the correct answer for this question. Move to the next question, number 132. Albert stain is used for option A, Staphylococcus species, B, Corynebacterium diphtheria, C, Clostridium perfringens, D, Clostridium tetani. Fact available for this question, it is Albert staining technique is a type of special stain technique. Since it is used to demonstrate a special structure in bacteria, it is chiefly used to demonstrate metachromatic granules found in Corynebacterium diphtheria. This bacterium is responsible for the disease diphtheria. So we can say from the given information it is Corynebacterium diphtheria, for which Albert stain is used for the staining purpose. So option B will be the correct answer for this question. That's all for the today. Thank you for watching the video. Please subscribe our channel LabTech World, and if you like our work, then like and share our videos with your friends who are preparing for the lab technician competitive exams. Thank you very much.